this week and next week, and we're done. I don't hear any joyous exclamations. There's one, okay. It's a different kind of meeting than I expected. All right, so we are moving into translation. And why do I have, that's translation. I've got the wrong thing here. I've got one last thing I want to say about transcription very briefly, because I overlooked it inadvertently last time. And then we can talk about translation. And that was having to do with transcription initiation in eukaryotes. I didn't look at my notes carefully when I was speaking last time. So I want to say just a little bit about the initiation of transcription in eukaryotes, because I've alluded to the fact that it is, in fact, a much more complicated, much more involved process than it is in prokaryotes. And I will talk more about this later when we talk about gene expression as well, because there are some considerations relative to the moving away and the clearing out of the histones that I won't be talking about here that will come up later. This figure shows the sort of process that is involved in the transcription of many eukaryotic genes. I'll remind you that in eukaryotes, RNA Polymerase II is the polymerase that synthesizes the messenger RNAs. And the polymerase is assisted in the initiation of transcription by numerous proteins called transcription factors, which I've mentioned before. And they have a series of letters that allow that are each distinguishing each of those individual factors. As I said, this isn't true for all eukaryotic genes. We see that there is quite a variety of ways in which transcription occurs in eukaryotes, but this is a very common one that we see. When we look at the transcription initiation in eukaryotes, by the way, these have abbreviations. So wherever you see the letter that's there, like you see this D, it would be preceded by the letters TF2, Roman numeral 2. Okay? So TF2-D would refer to this protein, TF2A would refer to this one, TF2B, et cetera, et cetera. Now, I don't put this on the screen to give you a lot of things, a lot of different proteins to memorize. That's not the point. The point is to show you very visually to start that this process is clearly, even with this very relatively simple scheme here, is clearly much more involved than it is in prokaryotic systems. I referred to the TATA box that I said wasn't always present in front of eukaryotic genes, but it was in front of genes that were expressed at high levels. And that sequence is a necessary sequence for binding of this factor here called TBP. TBP stands for TATA-Binding Protein. And in conjunction with TF2D, it is the first thing that binds to the promoter region of a eukaryotic gene. So TF2D and TBP together bind to facilitate, ultimately, the binding of the polymerase. You'll notice that the polymerase isn't binding. The polymerase binds only after some of the other factors, including A and B, have bound. And then after the polymerase has bound, other factors, including F, E, and H, all bind. So we have a complex of proteins that are located at that transcription initiation site. One of the requirements for transcription to proceed in eukaryotic cells is that the tail of the RNA polymerase II must be phosphorylated in order for the transcription to proceed. Okay? After that phosphorylation has happened, most of the factors either fall off or are left behind. The polymerase, in conjunction with TF2F, then moves forwards, as you can see here. Okay? Now, each of the proteins have functions. I'm not going to go through those with you. I don't think it's really necessary for our purposes that we understand each of the functions of those proteins. And like I said, this is really here to show you the complexity, the fact that it is a complex process that's leading to transcription initiation in eukaryotic cells. Okay. This shows that complex between the TATA-binding protein and the DNA, the TATA-binding protein being up here, the DNA being here. And you'll notice something that is something like we saw with the restriction enzymes, and that is the bending of the DNA duplex as a result of binding of this protein. Yes, sir. Question?
Was the tail on the
That's a part of the polymerase II. That's a structural part of the polymerase II. It's not part of the RNA. That's correct. It's a structural part of the RNA polymerase that's being phosphorylated. Okay? Which means at some point it has to get dephosphorylated as well. Yes? Is TBP required for TF2D to bind? No, because TF2D, they bind together. I'm sorry, I was thinking RNA polymerase. They bind together. Yeah. Yes? It's my understanding that TBP is actually a part of TF2D. It's actually a functional part of TF2D. Okay, so that finishes the last of what I wanted to say about transcription. So now we can turn our attention to talking about translation. And I apologize, I shifted the dates, which is why you see the translation showing here when it really should have been moved down there. I'll fix that later. But there's the translation. Oh, now I've said it wrong. The translation should be down one more. In fact, it's right here, so it should be there. Translation. So translation is the process whereby cells convert the information that's found in messenger RNA into a sequenced protein, or a protein with a sequence of amino acids. That sequence of bases in the messenger RNA, of course, leads to the, it is interpreted by the ribosome in terms of the genetic code to put in the appropriate amino acids in the right sequence in order to make the final protein. Ribosomes are fascinating, some people call them particles. I guess that'd be a way to describe them. These particles are complexes of about 50 or so, 50 to 70 proteins. They also include, in the case of prokaryotes, 3 ribosomal RNAs. In the case of eukaryotes, 4 ribosomal RNAs. The ribosomes are defined in terms of size. So somebody asked me in the term earlier, what does the S mean when we say 50S and 30S? And the answer is that S stands for Svedberg units, and they are measures of the rate of movement of these complexes in a centrifugal field. So an S is a measure of movement in a centrifugal field. Those that have a larger number will move faster in a field than those that have a smaller number. Okay? Those that have a larger number are therefore bigger than those that have a smaller number. So a 50S is bigger than a 30S, but as I noted in the answer I gave to the student earlier in the term, these numbers are not proportional. So if I said I had a 60S and a 30S, it wouldn't mean that the 60S is twice as big. It would just simply mean it's bigger. And a 60 would be bigger than a 50, but not necessarily proportionally bigger. Okay. What you see on the screen is a depiction schematically of the ribosomal units. There are 2 units, a 50S and a 30S. And since they're not proportional, neither are they additive. So if we look at the overall size of the ribosome here has a size of 70S. 50 plus 30 equals 70, right? Okay. Eukaryotes have very similar ribosomes, but they're larger than prokaryotic ribosomes. In the case of eukaryotes, that 50S is a 60S, that 30S is a 40S, and the overall size is an 80S. Okay? So we simply, if I say 80S, then you should recognize that I'm talking about a eukaryotic ribosome. If I say 70S, I'm talking about a prokaryotic ribosome. I'll describe the ribosomal RNAs that are in the ribosomes in a little bit, but I want to show you some features of the ribosome and the sort of picture of what's happening here as a sort of a guideline for us as we move forwards. We see on this picture in yellow and orange, whatever color you want to call that right there, that's the messenger RNA. And each of those circles is one individual base of the messenger RNA. The messenger RNA is being translated, and you can see that the messenger RNA interacts with 3 sites called E, P, and A. Now, as this ribosome is moving, it's moving from left to right. It is moving down the messenger RNA in a 5' to 3' direction. So DNA synthesis occurs 5' to 3', RNA synthesis occurs 5' to 3', and translation occurs 5' to 3'. So this guy is moving from left to right, which means that new sequences will appear in the A site first. Now the A site, the P site, and the E site excuse me, are places where the transfer RNAs will come in and bind and see if they can base pair with bases in each of these sites. If they can, and that test will be done actually in the A site, if they can, then everything is fine and dandy and translation proceeds. If they cannot, then they're basically kicked out of the ribosome until the right one comes in. Now, this is a pretty remarkable process if you think about it. We've got lots of possible tRNAs that can come in. There are 64 different codons, 61 of which code for amino acids. Okay? And it turns out there's not quite that many tRNAs for reasons I'll explain in a bit. But still, we have on the order of 20 or 30, we have about 30 or so transfer RNAs that could come in. So the likelihood that the one of interest is going to come in, if we just did it on terms of numbers, is 1 in 30. So that means 29 times something comes in, it's the wrong one, it gets kicked out. Right? And still, in spite of that checking and coming in and out, this process is still translating at the rate of 17 amino acids per second, which tells us a lot about how fast diffusion happens inside of a cell. It tells us a lot about that. Okay, so I'll say more about that process as we move along. The last thing I want to point out on this figure is you can see the protein that's being translated. That's this multicolored thing that's coming out the top. And the translated or the protein that's being made comes out a hole in that 50S ribosome. Comes out of a hole in the 50S ribosome. Folding of the protein can occur as soon as it starts to emerge. It doesn't have to, but it can. Okay? Okay. So that's the general features of a ribosome. This this figure on the screen shows how the transfer RNAs get their amino acid. Amino acids in a transfer RNA are linked to the 3' end of the transfer RNA. They are linked as a result of action of an enzyme, the long name, called an amino acyl tRNA synthetase. Let me repeat that. Amino acyl, ACYL, tRNA synthetase. I'm open for names. What? Amino acyl tRNA synthetase catalyzes the linkage of a tRNA. Oh boy, I'm telling you the wrong thing. This is actually doing translation. Sorry, I'm ready for the amino acyl tRNA synthetase. Let me go to that and I'll come back to this. Sorry, I'm showing you the wrong one. Where'd it go? Here it is. It's actually this process right here. Okay, sorry. Spaced out. All right. So there's the 3' end of the transfer RNA. Okay, right there. There's the 3 bases, C, C, A. And there's the amino acid that has just been added. Okay, so the addition of the amino acid was catalyzed by the amino acyl tRNA synthetase. Okay, is that clearer? Sorry. You guys want to call it something else? You want to call it amino acyl tRNA synthetase? Teri? T-E-R-R-Y? Okay, there's one name. Is it going to be Teri? Yes? Okay, so I'm actually getting to that. So I'll talk about the translation process in a sequential set of steps in just a bit. Okay, Teri it is, I guess. T-E-R-R-Y. All right, you're welcome to call that enzyme Teri. Okay. Anyway, getting back to where I was jumping ahead there. This is actually showing what's happening in the translation process. Here is a tRNA that has an amino acid attached to it. Here's an incoming tRNA, and this will get to your question in just a little bit. Incoming tRNA that has a different amino acid attached to it. These two become linked And what happens is that this linkage happens where this guy is put on top of this guy. Okay? So we see now that tRNA2 holds both of these. tRNA3 comes in, and this gets put on top of tRNA3. Okay? All right. And let me show you this in some more detail when we get to the ribosome. We've talked about accuracy with respect to transcription, and we've talked about accuracy with respect to DNA replication. Now we need to think a little bit about accuracy with respect to translation. And it turns out that cells actually have some proofreading that they do of translation, but they don't do it to nearly the extent they do with DNA or even with RNA. And the reason, again, is because proteins, it's okay to make a little bit of error in proteins. All right? So if we look at the relative errors that cells make with respect to the frequency of making an error, putting in the wrong amino acid, if they make an error of 1 in 100, okay, then that means if they have a protein that has 100 amino acids, then 36% of the time it's going to have at least one error. Okay? If it's 10 to the minus third, 1 in 1,000, about 90% of the time. Okay? If there you can see the numbers, et cetera, et cetera. So the probability of forming a protein with no errors depends on the number of amino acids and the frequency with which errors occur. It turns out that proofreading process is costly in terms of energy and costly in terms of time. Cells compromise and allow more errors to happen in making proteins in order to optimize the frequency, the rate with which the process occurs and the energetic considerations. Okay? All right. And to give you an idea, the error frequency is about 10 to the minus 4th, 10 to the minus 5th. That's about what it is in translation. So it's not real common in terms of errors. All right. The genetic code. The genetic code is something I think by this point in your schooling everybody knows something about. The genetic code is how information is stored inside of a messenger RNA. It is specified in units of 3 nucleotide bases, okay, that each of which can correspond to 1 amino acid except for 3 of them. So I'll explain that in a second. So to read this table, you see the genetic code starting at the 5' end of the codon we're talking about now in the messenger RNA. From the 5' end of the codon, the first base being U, the second base, in this case, being U, where my green pointer is, and the third base up here where this is a U also. So if I had U, 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 that would specify phenylalanine. If I had U, U, C, I would have phenylalanine. If I had C, U, U, I would have leucine. Now, as you look at this diagram, you can see that it has a lot of what we call redundancy, meaning that there's more than one codon that can specify a given amino acid. And you'll notice that it's not random, but in fact there's some organization. UUX, where X is any of the bases, I should have bad choice. CUX, where X is any of the bases, refers to leucine. If I said UU pyrimidine, I would be specifying phenylalanine. If I said UU purine, I would be specifying leucine. And you'll see, again, if you look carefully at this, that the split, like here where we have histidine or glutamine for CA pyrimidine, CA purine, they break along purine and pyrimidine lines for the most part. Okay? Now, why do we have redundancy like this? Well, it turns out that there are 20 amino acids that are specified by the code, and we have 3 bases. So 3 bases correspond to 64 possible 3-base sequences. You say, well, why don't we just have 2? Well, if we only had 2, there would only be 16 possible sequences. We couldn't get all of the 20 amino acids in there. We can't have 2.5 bases, all right? So that means that we have redundancy that's here. 3 of the codons correspond to stop codons. They are UAA, UAG, and UGA. Those are called stop codons. U, G, UA is a stop codon. Okay? UAA is a stop codon. UAG is a stop codon. Okay? That means stop translation. This is the end of the message. When the ribosome hits that, it knows that we've just finished making the protein. There's also a codon that's called the start codon. And the start codon is AUG. That corresponds to methionine. Methionine is always, or almost always, the first amino acid put into a protein. All right? First amino acid. So when you see an AUG at the beginning of a protein, that means that it's a start codon. But AUG also appears internal to a protein sequence. So just because it's an AUG doesn't mean it's a start codon. It's only a start codon if it's at the beginning of a coding region. Okay? All right. That's the genetic code. It's interesting when we look at the codon. So a codon, I'll repeat, just 1 second. A codon is a 3-base sequence that's in the messenger RNA. Make sure you understand that. The anti-codon is a complementary sequence that's in the transfer RNA. So you may remember when I showed you the structure of the tRNA earlier, I said that's the anti-codon loop. That anti-codon within that loop is a 3-base sequence complementary to the codon. Okay, let me take questions before I move on. You got a question, McKinley? So her question is, what's the significance of methionine in the middle of a protein? Simply the fact that, like any other amino acid, it's needed for structure of the protein. There's no significance per se, other than it'd be like saying, what's the significance of a glutamic acid? The cell needed, the protein needs a glutamic acid there to be functional. Okay? Yes, question?
Yeah, so how come the stop sequence instead of just like when the mRNA runs out?
Okay, so the question is, how come there's a stop sequence instead of just letting the ribosome fall off of the end? Okay? Don't have a good answer to that question, but we'll also see that the starting place isn't at the other end either. It's internal in both cases. It's perhaps the fact that it might be hard to hold on to that sequence at the very end, that there's not really more of a tail to it. That's my guess, but I don't know the answer to the question.
So there might be more, if it just fell off, there might be more transcription.
Well, that's true. That's true. I mean, if you think about it, that transcription in some cases isn't stopping at an exact place. So the transcription stop site is also a consideration, and you're exactly right there. Okay? Good observation. Okay, well, so I've told you that the codon and the anti-codon pair during the process of translation. Now, interestingly, when we look at the anti-codon, we sometimes see the base inosine. This occurs as a result of a chemical modification. Remember I told you that tRNAs become chemically modified. So sometimes the base and the anti-codon is modified to become an inosine. Why in the world is an inosine placed in the anti-codon? Well, it's done for efficiency. Why? Well, look at this. It turns out that inosine is able to pair with 3 of the 4 bases. It doesn't care. It's indiscriminate in its pairings. Well, why would that be useful? Let's think back to the genetic code. Where I had leucine, I could have CUX, where X was any of the amino acids, right? So if I had CUI, I could make one tRNA that would pair with any of those bases in the codon of the messenger RNA. That means the cell has to make fewer tRNAs. By putting that inosine at that 3rd position, it saves having to make individual tRNAs, one that would pair with U, one that would pair with C, and one that would pair with A. So that's for this reason that cells do not need 61 different tRNAs to code for all the different possible pairings with codons. Okay? And it turns out cells have on the order of about 20 or 30. They don't need all the ones that they've got. This also reminds us of something I told you earlier, that is that G can pair with either U or C. G can pair with either U or C, which means that U can pair with either A or G. So that also allows some efficiency in terms of making the number of tRNAs that are necessary for a cell to have. Okay. Questions about that? Yes? Does this affect mutation rate? Well, let's think about this. Mutation rate refers to the sequence in DNA. This is not in DNA, so it has nothing to do with mutation rate. Okay? It could have something to do with error rate of making a protein to a limited extent. So I think that maybe that's what you meant. Yeah. Okay. And you don't care about that. All right. Let's take a look now at the tRNAs and the pairings that they can exhibit. So this is now showing the anti-codon and it's showing the codon. We read the codon from 5' to 3'. Okay? So if I said, what is the sequence of the codon here, you would tell me it's GCC. If I said, what's the sequence of the anti-codon, I hope that you would tell me it's IGC, because we're reading from 5' to 3'. Right? We've got anti-parallel setup that we have in this case. Okay. So the anti-codon and the codon are anti-parallel, just like we always see nucleic acids. They're always anti-parallel. Okay. This now, again, schematically shows the structure of a tRNA. And I'm not showing you this to wow you with anything here. They're just to remind you of the structures that we talked about before. There at the top is the CCA sequence where the amino acid gets attached. It turns out that, remember, this is ribose. That is the sugar that's in ribosomal RNA. Ribose has both a 3' hydroxyl. It also has a 2' hydroxyl. And some amino acids get attached to the 3' hydroxyl, and some of the amino acids get attached to the 2' hydroxyl. I'll say a little bit about those in just a bit. Okay? So the amino acid doesn't have to go on to the 3'. In some cases, it actually goes on to the 2'. We see the amino acid up here. We see the anti-codon way down here. Now, this is kind of a cool thing. Right? Well, what's so cool about it? Well, what you see on the screen is the way that we always drew transfer RNAs, because we knew that this base pairing occurred, this base pairing occurred, this base pairing occurred, and this base pairing occurred. We knew that, okay? So we said, well, this is the most logical structure that it should have. And in 2 dimensions, this is the most logical structure that this guy can have. All right? But there's a problem. There are enzymes, one of which I've already given you the name Teri, that put the amino acid on up here, but in order for them to put the amino acid on up here that's the right amino acid, they have to be able to read the anti-codon down here. You say, well, so what? Don't they do it? Well, they have to do it simultaneously. It can't go down here and say, okay, and then let go, and then hope it grabs the right one up at the other end, because that wouldn't be any good. It's got to grab them simultaneously. However, the distance from here down to here is larger than the size of most of the Teri enzymes. There are, in fact, 20 Teri enzymes, one for each amino acid. Each Teri enzyme will recognize multiple different possible tRNAs as long as it has the right amino acid. But the size of this is bigger than some of the Teri enzymes. So how in the world does the Teri enzyme accomplish that? Well, the answer to this question became quite obvious after people looked at the shape of this guy in 3 dimensions. When they looked at it in 3 dimensions, they discovered it's not a flat, planar structure where this guy is way up here at the top and this other guy is down here at the bottom. In fact, it's bent so that it's more like this. There's the terminus, and there's the anti-codon loop. And this distance is about the size of a Teri enzyme. So it reminds us that two-dimensional representations are easy for us. And by the way, all those base pairings are still occurring. You're just not seeing them because it's very clearly because in 3 dimensions it's hard to envision these guys. Okay? But this structure reminds us that a simple 2D representation is good for illustrating what's there, but it's not, shall we say, anatomically correct. Okay? Okay. There's that general structure, blah, blah. Okay. There's another view of it. That's with more of a 3D consideration. And now it's showing you some of those base pairings that we saw earlier. Okay? Again, a little harder to envision looking at it in 3D. Okay. Well, now I want to turn our attention to that Teri enzyme that I tried to talk about earlier. The Teri enzyme catalyzes the reaction that you see here. Amino acid plus energy in the form of ATP plus a tRNA plus water gives, why does it say aminbo? It should say amino acyl tRNA, meaning that the amino acid is now covalently attached to the tRNA. And notice that we make AMP. We're splitting off a pyrophosphate off of this guy in doing this. Okay? This reaction, as I said, there's one Teri enzyme for each of the amino acids. So there are 20 different Teri enzymes. And by the way, I'm saying Teri, the people in the eCampus class will probably wonder a little bit about that, the amino acyl tRNA synthetase enzymes. Okay? So that's important to understand. Now, it turns out there's something kind of interesting and curious about this. This amino acyl tRNA synthetase is very unstable. It's very unstable. It's floating around in an aqueous environment, and it's the half-life of that bond that holds the amino acid to the tRNA has a half-life of about a second. It comes apart really quickly. That means we can't let it just floating around in water. And it turns out that as soon as this guy is made, it's linked to another protein, okay, that carries it and covers up that bond so that water can't get in there and break the bond. Yes? The product is what's unstable, the amino acyl tRNA. Good question. So that amino acyl tRNA is unstable. And it turns out that amino acyl tRNA is joined in E. coli to a protein called EFTU. We'll talk about that in a bit. EF stands for Elongation Factor. EFTU, to tell you the importance of that protein, it's covering up, it's protecting that bond. Its importance is so great that it's the most abundant protein in E. coli. Okay? You don't want to have any time waiting on an EFTU protein to show up to cover up that bond. You want to have so much of it available that it's there when it's needed, and it is. We have similar proteins in our cells that function in the same way and for the same purpose. Okay, so there is the bond, again, right there. Okay, let's see. Now, what this figure shows us, we're now looking inside of a Teri enzyme, the amino acyl tRNA synthetase. We're looking inside of this enzyme, and we're seeing the place where the reaction is being catalyzed to add that amino acid to the transfer RNA. And we can see that there are two sites. There's an activation site and there's an editing site. The activation site is where the catalysis occurs to make that linkage. And after that has happened, this flips over into the other site to be edited, meaning that that's where the proofreading is actually occurring. If the enzyme determines that, in fact, it has the right amino acid there, it will pass this guy off to EFTU. If it has the wrong amino acid in there, it will cleave it and try again. So there actually is some proofreading that's going on, and it's occurring at this step in the process. Okay. That's a really ugly picture, isn't it? My only reason for having this picture is, again, to show you the blue is the enzyme. So the blue you can see is interacting with and covering up here. This has an activation site. It's also got, that's where the CCA is sticking into, is in that activation site. And down here, the other end of the enzyme is reading this guy and making sure it has the right sequence of the anti-codon. If it reads this sequence wrong and puts the wrong amino acid on, then we're screwed. Translation isn't going to put the right amino acid in. Okay? So reading of this sequence is really critical. In fact, it's at this step, the process of putting the amino acid onto the tRNA, that translational fidelity is actually occurring. Okay? We'll see one other place where it occurs, but this is very critical. If we don't get this right one on here, then no other process we're going to do is going to make any difference. This process has to go as accurately as we can afford to do it. I won't mess with that. Okay. Now, I said that some Teri enzymes, the amino-acyl tRNA synthetases, some Teri enzymes put amino acids onto the 2' end of the transfer RNA, and some put it onto the 3' end. It's both the same end, right? It's the same end of the enzyme, but it's whether it's the 2 or the 3, which side of the ribose it's putting the amino acid onto. This shows us that there are two classes of amino-acyl tRNA synthetases. Class 1 enzymes shown here, there are 10 of them, and the class 1 enzymes all put the amino acid onto the 2' end of the tRNA. The class 2 enzymes all put the amino acid onto the 3' end of the tRNA. Okay. Now we're moving along well. Now, I show you this figure to show you something that is something to give you some perspective. Okay? Now, as you look at this, you see that it's color-coded. And you see you've got the 50S subunit of the ribosome here. You've got the 30S unit of the ribosome over here. And I've told you that there's about 50 proteins, and there's 3 different ribosomal RNAs that are found in the total overall complex. Okay? In the 50S subunit, the yellow guy is ribosomal RNA. Ribosomal RNA is a big honking part of that ribosome. So we look in the 30S subunit, okay, the green is the ribosomal RNA. So the ribosomal RNA is not some trivial piece of a ribosome. It's actually a pretty significant chunk of the ribosome. Okay? And the ribosomal RNA provides some very, very important functions, as we shall see. One of the functions that one of the ribosomal RNAs plays is one of those ribosomal RNAs actually catalyzes the formation of peptide bonds. That means it's a ribozyme. It's not an enzyme, it's a ribozyme. All right? We talk about the early Earth, where we think that the earliest genetic information was RNA. We also think that the earliest catalysts were RNA. And this is really good evidence, of course, that RNA can, in fact, catalyze reactions. It's catalyzing the most important reactions that's going on inside of cells, which is the making of proteins. Pretty cool. Okay? That enzyme that catalyzes that making of proteins is the 23S. ribosomal RNA. It's found in the 50S subunit. Okay? All right. So, pretty cool. Ribosomal RNAs are interesting and unusual. Okay? This shows a two-dimensional representation of one of the ribosomal RNAs. Ribosomal RNAs have elaborate, what's called secondary structure, where they have interactions between sequences that are close to each other. And we can break this down into 1, 2, 3 different regions: a blue region, a red region, and a green region. And in 2 dimensions, this is what it looks like. And down here at the lower right, that's what it looks like in 3 dimensions. Again, that 3-dimensional representation is anatomically correct, but not quite as easy to interpret as this representation is up here. And again, all the base pairings that you see up here are also present down here. They're just more scrunched up. Now, I always like to think about this as well, because we think of the process, and we've spent a fair amount of time last term talking about protein folding. And I hammered into your heads the importance of protein folding for protein function. It had to fold properly. If it didn't fold properly, we might get a prion or something like that. And this tells us that the same is true for RNAs as well. That this guy has to fold up, just like a protein has to fold up, in order for it to function properly. And so 3-dimensionality is not something that's unique to proteins. It's also very important for RNAs. Now, the same thing doesn't happen with DNA, because with DNA, we have a double helix. And the double helix doesn't fold. It doesn't have the flexibility that the single helix does of an RNA. I notice this is a single helix. Okay? A single helix. Here's nucleotide number 1, and you can trace it all the way around, all the way around, all the way around. And you get over here, and here's nucleotide 1542. That's one strand of ribosomal RNA that's going all the way through there. Okay. Now, there's another thing we have to consider, and it's related to the question I had earlier about why methionine, what's the significance of methionine in the middle of a protein? And my answer was that it was necessary for the function of the protein, but I wasn't telling you the complete truth. It turns out that there's something special about the very first methionine, at least in prokaryotic cells. The very first methionine does not go in as simple methionine. It goes in as what's called formyl-methionine, or F-met. Meaning it has a formyl group, which is shown in red right there, that's been attached to the amino end of the methionine. Why does that happen? Well, it turns out that during the process of translation, the incorporation of the very next amino acid, okay, because this guy is flopping out here as it is, the incorporation of the very first amino acid causes potentially an unstable structure to form if that is a free amine group. It's reactive, and it will bind, and translation will stop if the unstable structure is allowed to form. So to keep this unstable structure from forming, cells put a formyl group on the methionine that's on the initiator tRNA. The initiator tRNA is the first, it's the amino acid that contains that first methionine. The initiator tRNA has a slightly different sequence than a tRNA that incorporates methionine in the middle of a protein. Well, how can it have a different sequence? Well, it can. It has the same anti-codon, but that doesn't mean that the rest of the sequence is identical. So it's because of this difference in sequence that the cell can say, here is a initiator tRNA for methionine. Here's a non-initiator tRNA for methionine. I'm going to formulate this one. I'm not going to formulate this one. Does that make sense? Okay. That formulation happens only in prokaryotes. Eukaryotes have a different way of handling that reactive amine group. They have a different way of handling that amine group. Prokaryotes don't have that issue. Now, this is only a problem for the very first one, because the very first amino acid is the only one that's going to have a free alpha amine. All the others are going to have the amine reacting with the carboxyl, and it's going to cover it up. So we're not going to have that for anything except for the very first one. So that's why this formyl group is added. Okay. Now, the last thing I'll say today actually relates to what you see on the screen. Okay? When we looked at promoters inside of prokaryotic or ahead of prokaryotic genes, we saw that there were some common sequences that were present. If we look within the messenger RNA, we're not looking at DNA here, we're looking inside of the messenger RNA, and we line up the start codons, and by the way, you see a GUG there, I'll explain that in a second. We see that near that start codon, there's a sequence that is AG rich. Okay? This sequence has a name. It's called the Shine, S-H-I-N-E, Dalgarno sequence, D-A-L-G-A-R-N-O. The Shine-Dalgarno sequence is what the cell uses to recognize that that AUG is, in fact, the starting AUG. Otherwise, it wouldn't have a way of knowing. Okay? Now, I'll tell you next time how that occurs. It says here it pairs with a 16S ribosomal RNA, and it does, and I'll show you how that happens. But this Shine-Dalgarno sequence, the core is this guy right here, G, G, A, G, G. That is the part of it that most commonly appears, and there you see it right there, G, G, A, G, G, that most commonly appears. But you see variations on that G, G, A, G, G throughout here. The Shine-Dalgarno sequence is necessary for lining things up properly inside of the ribosome. For the last thing we need to do today, I need to ask you to stand. Everybody to stand. I need you to place your hands on your heart, because we're going to sing the Molecular Biological National Anthem. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome, translate mRNA, initiate and translocate from start to UGA. Okay, see you guys on Wednesday.